I'm here today with Humphrey Rang. It's a great pleasure to be here. Humphrey, of course, an eminent pharmacologist, ex-president of the British Pharmacological Society, and famous for his book, which I gather these days creates students coming up to take many selfies with him. So, Humphrey, I suppose what's going to be interesting for people is to know at the outset, what stimulated your interest in science? When did you know that science was something that was going to be for you?
Oh, I think I was always a pretty geeky boy, much more interested in science than in literature, for example. And I did science A-levels at school and was really switched on by chemistry at that time and biology. This was in the early 50s, and biology at that time was really a very descriptive thing. There was really no genetics, no cell biology. And it was all about the life cycle of the liver fluke and so on and so forth, which I found a bit tedious. So I wanted to go to university to do chemistry, but I was told firmly by Cambridge University that I wasn't clever enough for that. So I decided to apply to University College London, and they said, well, we don't have any places for you in chemistry, but had you thought about doing medicine? Because at that time medicine was really what you did if you weren't very clever. And so I went to UCL Medical School, and it was actually a revelation. The science that was going on in physiology and pharmacology was really quite outstanding, and I just got the bug.
When you were young and you described yourself as geeky from the outset, were you drawn to sort of the theory of science, or was it sort of, I mean, were you a practical person who liked to sort of fiddle with chemistry sets and whatever?
I did a lot of that sort of stuff, and actually set a few things on fire, and but I was interested in the theory as well. I think in physics and chemistry in particular, because they just seemed to have a rather clear-cut theoretical basis, which certainly appealed to me.
So you find yourself at UCL doing medicine.
Yes.
Was that scientific?
Well, it was, because I really got to know a lot of the physiologists and pharmacologists even as a second-year student. I used to drop into their labs and things like that, and they were always very, very welcoming and nice to me. And I did an intercalated BSc in physiology, and then went on actually to do a year's research in pharmacology with Heinz Schilt, which kind of set me off on the pharmacology track. So UCL was a very inspiring place for me at that time.
But there were many famous physiologists and pharmacologists there at that time.
There were, yes.
It'd be interesting, I think, to sort of hear, what it was like to be a medical student, and I guess in a relatively small class in those days.
It was about 60, I think, the class size, yes.
So you knew these people, and they welcomed you into the lab, did they? Exactly
so. I mean, one of my earliest recollections, going just on the introductory week at UCL, we were all invited to look round the various research labs in physiology and biophysics. And I remember going into a lab that was run by A.V. Hill, who was a very, very distinguished Nobel Prize-winning muscle physiologist. And there was this really tall, elegant, white-haired patrician figure in his lab, which was a rather surrealist place because it had a large number of umbrellas suspended from the ceiling over each bit of equipment, because the physiology classroom was in the floor above and used to flood regularly. And the equipment itself was housed in rough old orange crates. And this Nobel Prize-winning physiologist explaining exactly what he was doing, and this impressed me a lot at the time.
Amazing, really, though, that as an undergraduate student, you could just walk into the lab of a Nobel Prize winner and who would explain what he was doing.
Exactly, yes.
Inspirational, actually.
Yes, and Bernhard Katz was another individual, and he too was, very welcoming to, us 18-year-olds.
So in your intercalated BSc, was the topic pharmacology?
It was one of the topics. We had a series of options, and pharmacology was one of them, which was run by Heinz Schilton. Again, he was a very quantitative, analytical, clever man. A dreadful teacher, actually. His lectures were largely incomprehensible. But as an individual, if you actually got talking to him, he was extremely impressive. And so I learned a lot from him.
Was that your first sort of inkling that pharmacology was the thing for you?
He was my mentor, and as I went through the clinical parts of the course, I used to talk to him quite a lot. He had told me, look, don't drop medicine, just carry on in the clinical course. You may not be very good at it, but just do it. So I just did it. And at the end of that time, I wasn't really quite sure what I wanted to do in life. And I could have done a house jobs and proceeded in clinical medicine, but the opportunity came up for a research fellowship in Oxford in pharmacology there, which just seemed too good to pass up. So I went straight from UCL to Oxford.
What was it about pharmacology that you thought, yes, actually, this is for me, rather than physiology or biochemistry or the other choices? And then, maybe a second question, which is, was completing a medical course helpful to you?
As I went through medical school, I was very impressed by the ability of clinicians to diagnose and understand disease. The pathology was all, a lot of progress had been made. But it did seem to me that medical treatment still had an awful long way to go to meet up with the expectations. And at that time, antihypertensive drugs, for example, were really in their infancy. It was ganglion blocking agents, and just around the mid-50s, adrenergic neuron blocking agents had come in. These really made a huge impression medically because, patients with severe hypertension, as you well know, it was a really dreadful prognosis, and they wouldn't live very long. And I'd learned all about the physiology of blood pressure control and the cardiovascular control and so on. And it seemed to me that there ought to be far more opportunities for effective therapeutic intervention that had actually been exploited. So I think that general feeling that medical treatment was really trailing a long way behind the understanding of disease, that I thought that pharmacology needed to be given a push.
Right. Well, that was right, I think, wasn't it?
I think certainly, it was right, as it turned out. Yeah.
So off to Oxford to do research.
Yes.
And what was it you wanted to do?
With Heinz Schilton had worked a lot on receptor mechanisms. The whole theory of competitive antagonism, the stuff that's now bracketed in under the label of receptor theory, is something that Heinz Schilton had really pushed a long way forward. And at that time, Bill Payton in Oxford had also begun to get involved in trying to understand receptor mechanisms, and had published a paper in the Proceedings of the Royal Society about a thing called the rate theory of drug action, which turned out to be totally wrong, but was a very, very clever idea and intervention. And when I went to Oxford, I discussed with Bill Payton, and he was my supervisor. We thought that trying to get a better understanding of what receptors really were, as opposed to just abstractions, algebraic abstractions, which is really what they were at that time. And we thought we'd try to get a handle on it. And at that time, the availability of tritium-labelled compounds was just coming up, and liquid scintillation counting had been invented, and you could buy liquid scintillation counters. So we started off using tritium labelled atropine to label muscarinic acetylcholine receptors in smooth muscle, where an awful lot of the quantitative pharmacology had been done. So we had some numbers to fit with the binding experiments. But it turned out to be a nightmare project, actually. Everything went wrong that possibly
Most projects
could.
do turn out to be nightmares at some point, don't they?
They do, yeah. So I worked for a good 2 years on this, getting more and more in despair about whether we were ever going to get the thing to work. But eventually it did work. And so we were able to label muscarinic receptors convincingly in guinea pig ileum smooth muscle. That was a breakthrough at the time, because it was really the first notion we had about, these things are actual biochemical entities, not just abstractions. And so that was really a...
Can you remember how you felt when you first realised you got an answer to that question?
I just felt bloody relieved because I needed to write a thesis.
And at UCL you describe the people around and the importance of actually a cohort of people.
Yes.
What were at Oxford at the time, and who were the other people that were there with you, training with you, postdocs and so on, that were influential?
Well, the department was amazing in Oxford. There was Bill Payton, who'd really... He was one of the pioneers of clinical pharmacology, actually, and still had a big interest in clinical applications. And Edith Bulbring was there doing pioneering work on smooth muscles. Hugh Blaschko, who did a lot of early work on monoamine oxidase inhibitors, a biochemical pharmacologist. And these are refugees from Nazi Germany. Miles Vaughan Williams was there doing his work on antidysrhythmic drugs. And particularly important for me, there was an organic chemistry group within the pharmacology department doing pharmacological chemistry. And I spent a long time talking to Edward Gill, who was a very, very clever organic chemist. When we'd got atropine to label successfully, we decided that we'd try and make a covalent ligand using the same kind of principles. And Edward was the kind of person, if you chatted to him over a cup of coffee and said, well, could we possibly, do you think, manage to bind covalently to muscarinic receptors? And he'd scratch his head and go away, and 2 or 3 days later would arrive with some white powder and said, try this. And so this was extremely fruitful, actually, and we did get a specific covalent label. And that too was a big help in understanding more about what receptors were. I was no biochemist, and the protein chemists in the biochemistry department in Oxford were really not interested in this kind of thing, because we'd shown by the labelling how scarce these molecules were compared with the kind of things that biochemists like to work with, and so they thought they would be wasting their time trying to characterise it biochemically. And we had a fairly amateurish go at it. It was clear that it was a protein, and that was an advance at the time, because there were all kinds of theories about whether receptors were basically lipid in origin. So we were able to show that it was clearly a protein, and we got some very, very rough molecular weight estimates, but it needed a really good committed protein chemistry effort to advance that, and we didn't have the resources to do that. But other people then did. I mean, Arnold Burgin's group at Mill Hill did it in a proper biochemical way instead of an amateur pharmacological way.
So that sort of what actually was a rather profound discovery, which you describe as a relief, but in fact was the basis then of your PhD.
Yes, it was.
And
yes.
Took you, I guess, on a path to pursue that in postdoctoral studies and beyond.
Yes, I moved off in slightly different directions. We did a lot of studies on desensitization, also on channel block. I mean, because Bill Payton's interest in hexamethonium as a ganglion blocking agent, which he and Eleanor Zymis had looked at this methonium series and found decamethonium and hexamethonium and a number of others doing interesting and different things to nicotinic acetylcholine receptors. We decided to have a go at the mechanisms involved there. And I mean, Bill Payton had always puzzled about why decamethonium and hexamethonium were so different in their pharmacology. And we did a lot of studies on using electrophysiological techniques on ganglia and on neuromuscular junction, and pinned down some of the reasons for these differences. And in particular, we found that hexamethonium was really not a receptor blocker at all, which was what it had been designed to be, but was actually a channel blocker, and this accounted for a lot of the properties, and the fact that hexamethonium is a bit smaller than decamethonium allows it to get into the channel of ganglion cells where decamethonium couldn't. So it explained a lot of the pharmacological difference. So I got very interested in this. It's a fairly kind of esoteric corner, and of course now none of these drugs have any particular interest, so it was a bit of a cul-de-sac, but it was very engaging at the time.
Well, and actually it became the era of people trying to look for channel blockers and receptor blockers and so on, and that whole area became very profound for the pharmaceutical industry, actually, didn't it?
Yes, it did, that's true. One of the things that we and other people were working on at the time was the relationship between channel opening and channel block, and we and others were able to show that you had to open the channel in order for the blocking agent to get in, and then the channel closed and it was stuck there. And so these sort of details about which became part of the theory of use dependence of channel blocking drugs had quite important implications for cardiovascular.
Well, it still does. People are talking about use-dependent blockade of channels as a problem to try and solve, so it hasn't gone away as an area. What sort of size group did you begin to have working with you, and where did funding come from for that sort of thing?
The original project, I don't think, got any funding apart from departmental funds, because the whole grant business was just not as prominent in research economics as it is now.
Certainly wasn't a measure of success.
No, absolutely not. And so the PhD work was done. I don't know. Bill Payton produced the money from somewhere. I never really discovered where. And it was just me. I mean, I didn't have any help.
And from Oxford, you then moved?
I stayed in Oxford for about 10 years, and I spent a year's sabbatical at the Albert Einstein College of Medicine in New York, working with Murdoch Ritchie. That too was a really engaging and interesting year. And I came back to Oxford and got a college fellowship at Lincoln College as medical science tutor there. That was very hard work, but very enjoyable. And I could have stayed in Oxford, but I just knew somehow that it wasn't a lifetime career for me.
So you fancied the challenges of South London?
I actually moved to Southampton for a couple of years. They were setting up a new medical school there.
Oh, yeah.
And so I went as professor of pharmacology there. And they managed to they made a policy of taking on a lot of mature students to study medicine at Southampton, and they had quite interesting ideas about course structures and so on. I really found that these more mature students, there are a lot of lawyers and musicians and all kinds of people coming into medicine for the first time, and they just gave the place a kind of balance and maturity which conventional medical schools didn't have this same sort of balance, really. But research was much more difficult at Southampton.
Were you unusual in loving teaching and research? Because we often hear these portrayed as things that pull in different directions, but it seems to me, if I look at what you've done, you've actually brought the two things together.
Yes, I've always enjoyed and seen a lot of importance in good teaching, because I experienced a lot of good teaching, and I suppose in Oxford I felt that these very bright undergraduates didn't really need much teaching. They would have got on fine anywhere. Whereas at Southampton, they were a less privileged bunch, and I felt that there was more reward in setting them on the right track.
So from Southampton to Tooting?
Yes.
Where I met you, because I was an undergraduate in 1978,
right.
And
yes,
you were the
right.
professor of pharmacology at the time, with
yes.
a rather amazing young faculty, actually, of St George's at the time, I thought.
Yeah, George's was a very happy time for me. I spent 5 years there and developing the course and developing the department, and it's done very well, I think.
Some rather distinguished people came out of that department, didn't they?
Yes, well, I've worked for many years with David Cahoon, who joined me at St George's and set up a group there, and then I was asked to think about moving back to UCL after I'd been at George's for 5 years, and this too was very attractive, and David was able to come with me, so we were able to set up quite a good and lively lab there.
What would you describe as your sort of major scientific achievement during that period back at UCL, or the St George's to UCL period?
Well, that was really the time that we started doing a lot of electrophysiology. But I suppose, in a way, David, we worked together, but I think David was actually much more the intellectual driving force of that group, and I think the work they did on single channel characteristics and so on was among the best work that came out, and I was only very peripherally involved in any of that. And I also at that time was asked to become a consultant to Sandoz. That was really how I started to get a foothold in the pharmaceutical industry, and very interesting too.
So I do remember being spectacularly bamboozled by David Cahoon in my first ever intercalated BSc lecture on I think it was membrane noise at the time,
yes.
Which left me intrigued, but I think with a lot of work to do to try and understand it. When following that, when did the book start? When did the idea of the book start appearing?
When I went to UCL, because Heinz Schilt had retired as head of department, and there was an interim head of department, but Heinz was still around, and he had for a good many years been author of a book that was originally written by A.J. Clarke called Applied Pharmacology, which I'd actually used as a medical student. It's a very, very good textbook, actually. Heinz had been keeping this book going, I think, through 12, 13 editions. He wanted to give it up because he felt he was getting too old for it, and asked me to take it over, although it had been really good in its way. It had certainly got a bit whiskery and really needed a total revamp. So we just brought it into being as a completely new textbook. And there are still bits of A.J. Clarke's book still in there, actually, but not very much now. So,
well, it became a classic, of course. I think most people watching this have probably used it, read it at some point, and I know our new CEO got it as compulsory reading from me as a starting point.
Okay, good.
So you were at UCL,
Yeah.
juggling teaching, research, the book, and the politics of UCL, and decided to then become more involved with what was then the Santos Institute, I think, wasn't it?
Yes. Well, as I say, I served as a consultant with Santos for a while. And I'd always had this bug in my mind that drug discovery was a really worthwhile enterprise. And although I'd done a lot of quite basic pharmacology, I'd always had in mind the need to improve therapeutics. And so that really moved me on when I joined Santos and learned a bit more about the whole drug discovery, pharmaceutical industry environment. And then they decided that they would like to set up a research institute in an academic environment in the UK. And so they made a deal with UCL to set up the Santos Institute, and I was asked to run that, which I did very happily for 12 years, I guess.
And what did you learn during that period, in industry rather than academia, albeit in a slightly sort of different setting from normal industry?
I'd had absolutely no experience of the commercial world at all. And what struck me very powerfully, and I still does in a way, is the cultural divide in the industry between the science on the one hand and the commercial operations of a large company on the other. And the scientists seemed to me to be just like scientists anywhere.
Yeah.
I mean, they would run up and down the corridor in excitement if an experiment was going well, and they were just as committed to finding things out as academic scientists. But of course, where they were constrained was in the kind of things that they were supported to do. Now, there are a lot of very clever people in Santos, and I think a number of them realised that this rather dirigiste approach to research wasn't always very effective in making the kind of discoveries that they wanted to make and giving people a freer hand.
I mean, was that the experiment they were trying to do in having this institute in UCL?
I think it was. I think they deliberately had a very hands-off approach to allowing us to do whatever we thought. We were asked to focus on pain and finding improved analgesics, because there was really, still are, honestly, still only aspirin-like drugs and morphine-like drugs. And I certainly felt that there ought to be other ways of approaching this problem. So we started looking at sensory neurons and their characteristics. And we did a lot of the early work on capsaicin, and just using the conventional pharmacological techniques, it became very clear that there was a rather specific receptor that was responsible for activating sensory neurons by capsaicin. And we were not successful in identifying and cloning this thing, others were. But I think it was based on the pharmacological work we did, that made it clear that there was something to be looked for. And others then found TRPV1 and all the other trips and loads of drug discovery programs, none of which I think have yet succeeded.
No, we've had our own excitements around that. Yes. If I sort of look back across your career, let me ask you, what are the discoveries or the findings that you feel most proud of, that you think have made a difference in pharmacology?
Well, I think the receptor labelling really set off a substantial avalanche almost. I mean, the kind of binding studies that we took 3 years of struggle to do is the kind of thing you now do on a Friday afternoon. But I think, if I'm honest, I mean, the thing I take most pride in is the book. I've always felt, and I think increasingly I feel that whatever research you do today, somebody else would have done tomorrow. And so, it's jolly nice to add your brick to the research wall. But the thing that certainly made most difference, really, is the textbook and the way that it's switched a lot of people on to a better understanding of pharmacology. So I am very proud of that.
I think rightly so. And I think in terms of a new generation of people who've been inspired by pharmacology, I think that has been instrumental, actually. So, very, very distinguished career, elected to the Royal Society, and then you had a period as president of the British Pharmacological Society. I just wonder if you look at that period and say, what is it that you're most proud of?
Well, the British Pharmacological Society, like most learned societies, kind of started off as a nice cosy members club where they got together, they had dinner, they presented research papers and discussed it all. And it served a useful function. But it was increasingly being realised that the learned societies had another role to perform. And particularly when a lot of them became charities, they had to have a charitable function. So to promote the discipline in the outside world was important. Like a lot of the other conventional, traditional biomedical disciplines, pharmacology was tending to get overtaken by the new things, particularly molecular biology and cell biology and neuroscience and so on. And we thought that within biomedical institutions, pharmacology was being seen as a rather old-fashioned discipline which was no longer relevant or no longer as relevant. So one of the things that I was certainly involved in was setting up a project which is running now, actually, called Focus on Pharmacology, where we're doing, or the society I should say, is doing a number of things to find out exactly what pharmacology is being taught in higher education institutions, because there was really no good collection of data about how the subject was being taught in these various places, and also trying to get information from the pharmaceutical industry and other organisations who employ pharmacologists and the contributions they can make to the work of our environmental scientists, for example, environmental toxicology, sports medicine. I mean, there are so many areas in which drug action is important that it's better understood. And we have a lot of clinical pharmacologists in very significant, influential posts who are certainly going to play a role in this as well. So moving, as I say, from the cosy member society to a much more campaigning organisation, I think was an important change that I was certainly strongly behind.
And pharmacology really is an integrative discipline, isn't it? I mean, it brings together many other things to focus on the very problem that you alluded to earlier on, which is how do you improve therapeutics?
Yes.
And I think that remains a problem and remains something that society ought to be interested in.
Yes. Well, also how you stop people doing themselves harm with drugs.
Yes. So let me sort of draw this to a bit of a close and ask you the sort of question that's almost unanswerable, but it'd be interesting to hear your view, which is, if you put yourself back to a 21-year-old leaving university now, what are the problems you'd go for? What would you try and tackle as a emerging pharmacologist?
I really do feel that neurodegenerative disease is still a bit of a therapeutic wasteland. There must be good pharmacological approaches if we could only find them. I'm slightly unhappy about the huge amount of resource and focus on cancer. Of course it's important. I'm not saying it's not, but there's something like 7 or 8 times as much money being spent on cancer research as on Alzheimer's, for example. And given a choice, I'd rather have cancer than Alzheimer's, frankly.
You'd have a go at trying to crack that.
Yes, and I know plenty of very clever people are
Yeah.
committed to doing just that. But I think, well, just as with the pain research, you start something up and it either jumps all the hurdles that it needs to jump to get into therapeutics, or it fails at some stage along the way. But you mustn't be discouraged by that. And the more initiatives that are up and running, the more likely it is that somebody will jump all the hurdles.
Humphrey, it's been really a great pleasure talking to you, actually, and you taught me as a student, and I've massively enjoyed your book, which has been influential, and you've advised me in my current role when I first started off. So I owe you a huge debt of gratitude, and I know lots of people have been inspired by the work you've done. So it's been a real privilege to talk to you today. Thank you.
It's been a real pleasure for me. Thank you very much.